Start our class. And so welcome back. There are these internships I mentioned on Monday. I can answer questions about them, and they are good experiences, no matter what your future interests are, because they give you good science experience and analytical experience like that. Yep. So what you do this week, and so update this because some of the stuff was due on Tuesday, and so we took the quiz the second time. I haven't finished grading that, but first quiz we went back on that to try to keep learning and stack up our learning. So now we should watch the second quiz video, NSP video. That one is just 11 minutes long, and everybody should watch it and go back over that and think about it. Then I guess it's the 5th today, and so this Wisconsin to Boston drawing assignment is due today, and that's where we set the stage for understanding a little bit about oncogenic retroviruses and how could they be a problem and how did they help scientists identify some of the first oncogenes. And we're learning about RAS, and which our author studied. We're learning about MIC, and then next week there's this transcription factor video assignment, and so we'll talk a little bit about that in class today. And in that assignment, you're going to answer questions. So there are half a dozen questions that are like exam questions about different methods used to purify, identify, characterize transcription factors. And then we have this assignment. So we're going to talk a lot about the estrogen receptor. It's a more complicated transcription factor than MyoD because it needs the hormone estrogen. And anytime we're talking about the estrogen receptor, the same is true about the testosterone receptor. It's just male versus female in all vertebrates. So humans and all vertebrates on our planet use estrogen and testosterone as a sex differentiation gene, hormone rather. And so this is an assignment where I'm trying to give you opportunity to try some different types of learning. And so this assignment that's due a week from today is actually about half of it's a review, putting it into this learning style, and then about half of it is some more stuff that will add in more detail. So we're going to learn a lot about the estrogen receptor because it fits in really significantly in chapter 7, and then it's actually the beginning of our next chapter in chapter 16. Chapter 16 starts off with hormones, and it talks about estrogen receptor. The estrogen receptor and the testosterone receptor are members of a gene family, and it's a big variety of genes. In humans, there are 48 of these members of this big gene family, and so they are actually, it's referred to often as a superfamily because the members are doing such different jobs. And so there are just many, many different angles that we want to dive in and appreciate the estrogen receptor more. So we have a drawing assignment that will be kind of following up, kind of reviewing it, and kind of stacking up on that, like that, okay? And we're, going to have 3 chapters on our first exam, which is 2 weeks from Tuesday, and so next week we'll really get into that chapter. And so we're in good shape, but that chapter is one of the central chapters on our course. And so, we talked some about adding MIC, and why we plug these in is it illuminates and illustrates a significant concept, and that concept, where, when, and how much. And oncogenic retroviruses that have MIC are a good example of that. We talked about how all viruses are expressed at a very high level. They have very strong cis-regulatory regions in their genes. And if you connect one of those to the MIC gene, even though the transcription factor of the MIC gene is wild type, totally normal, we make too much of it and get cancer. And so that's part of chapter 7, talking about that, talking about regulation of gene expression. That's what that chapter is about. And so the clay model and everything we've been building up on that is part of that chapter. And now we're talking about, well, how do they get their job done? How do they actually do their job? And so, just think about how we're dealing with drawing, of a gene, and we can be like, okay, there's our gene, and so we've got the promoter and enhancer here, and understanding is that MyoD binds and somehow activates the myogenin gene. So this is about, well, how do they do that? And there are different ways, and we talked last time. Yeah, Don Diego.
So when you say MyoD, you mean with MyoD?
Yes.
Okay.
Yep. So here's the MyoD protein. It's a transcription factor. It's bound to the enhancer, the myogenin gene, and it activates its expression. So what does it do? And we talked last time about it could be far away. It could be, the enhancer could be far away from the start site. It could be in either direction. And so somehow it gets its job done by tethering, being tethered. And so it must be tethered, and somehow in being in this region, it must help the rate limiting step. And the first thing that it would make sense is maybe it would just grab this RNA polymerase and help RNA polymerase get to the start site, because that's really the action, is we want to get that going and get that transcribing the RNA. Like that, okay? And it doesn't grab the RNA polymerase, but it does grab another protein. And the what protein that it grabs is a protein that binds here, and there are a whole bunch of proteins that bind to this promoter, like 20, 25 proteins. And together with RNA polymerase, they make up this pre-initiation complex. And that's a nice name, pre-initiation. Initiation means when the RNA polymerase starts doing its job, starts copying the RNA. And prior to that, it's got to get, like, land there, find the right spot, and get all ready. And there's a bunch of proteins that help it do that. And if one of them's missing, it's not going to get done. And one of them's missing is a rate limiting step on the myogenin gene. And just to quickly show you an image of, I have it on here. So what we're talking about is the clay model videos, and we're saying there's the MyoD protein, and somehow this clay bands, and the MyoD protein helps something here. And what it helps is, in the clay model video, I added these pre-initiation complex proteins. It's just a bunch of little same color proteins, because assembling the pre-initiation complex is generally kind of a boring story. The big action happens here. That's what really matters. But we do have to get these things done. And in some examples, in an advanced course, there are changes in the pre-initiation complex, in different settings. But in general, the promoter has proteins that bind to it that are just helpers, everything getting set up. If we go to that analogy of the burger shack at the fair, the pre-initiation complex would be the morning when they're bringing it all there. And you're like, did you bring the stove? Did you bring the propane tank? Did you bring a spatula? Right? Lots of stuff has to show up even before the stand can open. Right? Did you bring the wrench to attach the propane tank to the stove? If any one of those is missing, the shop's not going to open right then. So one is missing here, and it's one of these proteins, and and so that's where we're at. That's where we're adding to the clay model videos. And the protein that's missing is called TBP. And so MyoD is going to physically bind to that TBP protein. Like the bitey dog is going to bite it and physically bind it. And so because of that, we have to add some information to all the transcription factors we're learning about. And that is, a transcription factor has a place that it binds to the enhancer. So there's the enhancer. And so this MyoD has two regions or domains as part of one protein. And one of them is called the DNA binding domain. And then it's this other part here that has to physically touch, literally physically touch the TBP protein. Somehow grab it, somehow bite it and bind it and have it. And what it's doing is really, it's tethering BBP to this area, getting it close to where it's supposed to be, helping it find its location. And that domain is actually called the transacting domain. And you can think about it as being like, oh, it's involved in transcription, so trans probably means transcription, and that's a good answer, right? transacting, transcription activating domain, because that's what it's doing. And the transacting domain, whatever transcription factor it's on, is going to affect the rate limiting step. So that's the action part of it, right? We got to get that there by binding to DNA and the enhancer, and now it's there and it's doing its job. And in the case of MyoD, it just binds TBP and holds it to help TBP get here. The actual official word for transacting domain means it's acting in trans. It's that old genetics term, where it's not part of the myogenin gene, but you need it for the myogenin gene to do its job. And it's acting in trans. It came from somewhere else. It came from a different genetic location. It came from the MyoD gene, like that, okay? So that's what we're doing, and it's just as simple as that. The rate limiting step for myogenin is it's hanging out there, ready to go, and it's missing this TBP, okay? So then, this is just a little drawing of what this looks like, really typically, is that the DNA will be bending, and it's just floppy. And sometimes there's a protein that helps it bend, helps it get in position to bring the enhancer near to the promoter and start. Because it's going to bind, it's going to do something at that location. So that's what we're really doing. The enhancer could be far away from the start site, but it's going to touch and hold something, do something at the start site, okay? So that's what we're talking about there. Here's just a drawing of some of the proteins in the thing, and our protein here is TF2D. And TF2D is a multi-subunit protein that includes TBP. So there are just lots and lots of proteins here. They got to get together, again, like all the things, got to get together in the morning before you open the burger stand, like that. And so you can see here that TBP has a really nice name for us. And it looks like this. So here's the start site where transcription unit begins, and people call that start site +1. It's the first nucleotide in the transcription thing. And so right next to it is the promoter. And the promoter has some different sequences that these pre-initiation complex proteins bind to. And lots of the promoters on our genes, like I think maybe 70% of the promoters on our genes have something people call a TATA box. And it's just the letters T, A nucleotides, T-A. And who binds there is TBP. And TBP stands for TATA binding protein. So as students, you're like, oh, yay, that's a good name for a protein, because it just means TATA binding protein. And in lots of genes, it's just hanging out. It's just there, ready to go. It's ready, it's ready to open up the burger stand. But in our myogenin gene, we have to have the MyoD transacting domain somehow bind to TBP and help it get in place. That's the rate limiting step. So we just do that, hold on, and go like that. Okay, that's it. So very physical, very commonsensical. That's it. Oh, I'm here with the propane tank, like that. That's it, and it goes and it increases the rate of transcription. Nothing else is needed. Everything's ready to go, and we just get going and copy. Okay, and so it's like that. And it's a very famous protein like that for this reason. It's a pretty boring protein, it's just got to get there and do its job. It's just part of the pre-initiation complex. There are some differences. I said not every gene has a TATA binding protein binding site, a TATA box, but many, many do. So it's a very common protein, and sometimes it's the rate limiting step. Sometimes it's a different rate limiting step of the pre-initiation complex. One of the other proteins is missing. Sometimes it's the RNA polymerase, or part of the RNA polymerase is missing. So we have this general idea of some transcription factors help assemble the pre-initiation complex. Oh, MyoD does that, that's how it does. And it's specific. In this case, it gets the TATA binding protein. That's the part of the pre-initiation complex that's missing, that's rate limiting, okay? And so that goes back to here, and we could zoom in on this and we make a whole nother clay model video up close, like that, and show specifically how the TATA binding protein does that. And we're picking on that as one example of how we increase the rate of transcription. And sometimes it's assembling the pre-initiation complex, RNA polymerase, and its helpers like TBP. Here's just an interesting fact to be aware of, the biochemistry of it. So you could say that MyoD increases the binding affinity of TBP for the Tata like that. And studies show this, that when MyoD binds to the binding protein, TBP, TBP actually helps change the shape of MyoD, and MyoD binds to the enhancer better. So it's almost like a handshake, a deal, a team like that. So that biochemistry is just, it's just fascinating to see in action, and it makes sense, and as long as you had time, you would understand that. It's a good example for what we want to talk about. And so that's the first example, right? And there are many, many genes that help assemble the pre-initiation complex. Some of them help bind Tata binding protein, some others, including the RNA polymerase itself. Our second example of a major way that transcription factors affect gene expression is that they release this arrested or paused polymerase. And so if we just go back to this slide, and we have this image of the pre-initiation complex assembling, RNA polymerase has not started copying, has not started transcribing, has not synthesized any of the RNA. Later, it starts. Later, it gets released from the pre-initiation complex and it starts going down the gene. And in many genes, the RNA polymerase has we got that far, and it's like that person that's handing out the burgers and they just hang on to them. It's already started, and typically the RNA polymerase has gone about 70 nucleotides down the transcription unit, and it just stops. And that's where many, many transcription factors affect the rate limiting step. It's like, hey, Donnie, let them have their burger, like that late, okay? And so that's our second one. And the example we're going to use is a good, really memorable example. And it's a new transcription factor, and it's called heat shock transcription factor, or HSF. And there are orthologs of HSF in every eukaryotic cell on our planet. So every cell in our body has HSF right now. So it's a good example for us to talk about. And every animal and every plant and every eukaryotic cell, like the yeast that causes bread to rise and so forth, like that. So HSF, a transcription factor like that. And so we want to learn a little bit about it, right? And it's involved in this thing called the heat shock response. And the heat shock response is like emergency first aid for a cell. And what happens is it could be caused by heat or other stresses that cause a lot of protein denaturation, okay? So you can heat up proteins and denature them. And so like at the site of a burn, right, at the very point of the burn, you probably have a lot of cells that die. But then there'll be some cells nearby that get heated up. They're not dead, but they got heated up so much, there's a bunch of proteins that got denatured, and the cell's gonna die if you don't fix that. And so the heat shock response is simple. The heat shock transcription factor activates a set of maybe about 10 genes, so a bunch of target genes, and they're called heat shock genes, so great name. So sitting in the enhancer of those will be this heat shock transcription factor, and those genes are all ready to go. What's their rate limiting step? They have an arrested polymerase. So this is an excellent example for us to think about, because this is emergency first aid for a cell, and you gotta make those now. So having them have an arrested polymerase as the rate limiting step is great, because you're just like go, 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 like that, right? What the heat shock genes do is they code for proteins that actually help refold proteins. And so they run around the cell, refolding denatured proteins and saving the cell. So the heat shock response is found in every eukaryotic cell. That's its function. It's super old, obviously, and it's emergency first aid for the cell. And you gotta make those heat shock proteins, and you gotta make them fast, okay? So it's a great example for us, because you'd be like, oh yeah, that makes sense. Those that have an arrested polymerase. And all we gotta do is get the heat shock transcription factor to do it, okay? So as part of this, we just want to talk about the regulation of the heat shock transcription factor, because it's got to get switched on, right? And it has to get switched on fast. So remember, we have a list of transcription factors and how they get regulated, right? And one of them we like is FOXO, it gets phosphorylated. One we like is ELK, it gets phosphorylated. One we like is the estrogen receptor, right? So how does the heat shock transcription factor get activated? And it's just a very, very sweet story. And it goes like this. So the heat shock transcription factor is always present in the cell. So that's HSF, and those are monomers of the heat shock transcription factor. So that's the heat shock transcription factor, and they are always present in every one of our cells. So we're always making this protein, but it's not active. And when it gets activated, it's going to bind to the heat shock genes. And the heat shock genes have an enhancer. And the heat shock transcription factor is going to bind to it. And that enhancer is called heat shock response element. So it's an enhancer, but people give it that response, because that's how they discovered it. That's how it guides, any gene that's connected that has one of those is going to get activated. So a heat shock response element. And so on these heat shock transcription factors, right, here's the protein, there must be a DNA binding domain. That's going to bind there, like that. And all of them have them, and they have a low affinity. They don't stick very well to the heat shock response element. And what happens, and we'll talk about it and redraw this, what happens when there is a heat or a stress is that now a group of 3 of these monomers join together to make a trimer, a trimeric protein. And so you know that some proteins do that. They bind together to make a dimer, trimer, multimer, like that. There's lots of proteins that have multiple subunits. And that's what it's like. Right now, the heat shock transcription factor is in our cells as a monomer, and it's not bound to the heat shock response element because it just doesn't bind very well. But then we have a trimer, and I always say that's like a 3-armed climber, right? Like if you're climbing up a rock wall and you have 3 arms, you're probably not going to fall off because if one hand lets go, 2 other hands are holding on. And so the affinity for the trimer to bind to the response element is really high. And so that's the trick of the switch, like that. And now we're just going to zoom in. We're just going to zoom in on the heat shock transcription factor to see what does that look like. And the answer is that the heat shock transcription factor monomer looks like this, where it's folded over like that. So that's HSF monomer. And this is an unstressed cell. And what's going on is that this, when it's folded, it's covering up the binding site where the transcription factors would bind together. So that when we have stress, when there's a stress, this protein itself gets, like you could say, partially denatured. It changes its shape. And it unfolds a little bit, and it exposes that trimerization domain. And then we have 3 of them bind together through non-covalent binding. And so that's the trick of it. And if you happen to know anything about how a house thermostat works, it changes shape at different temperature, and that's what switches on the heat or switches it off. So the heat shock transcription factor is like a little thermostat where If things get stressed, it changes its shape, and all that does is it changes it and exposes the trimerization domain. They grab and join hands, and that just makes it so they'll stick better. And that's a great thing because that's all that has to happen. You know, we don't need to make some other changes, phosphorylation or stuff like that. Just get it unfolded, and it'll bind with high affinity because it's binding like a three-handed primer. Like that, okay? So we want to add that and add that to our story of regulation and transcription factors. So this just shows a little visual of that, no big deal, and we add it to our list of transcription factor. So next one on our transcription factor list is there are plenty of transcription factors that get activated by binding together. And so it's very unusual for it to be a trimer. That might be unique. But it's very common, and we'll meet other transcription factors that need to form a dimer. And before they can do their job, before they're switched on, they need to join with another protein. So this is a good example of that. And it's just a memorable example that people like to use because of the story of heat shock and how heat shock is universal in eukaryotes. And then it gets activated. And what does it do when it gets activated? It binds to the heat shock response element, and it causes the arrested polymerase to be released. So we can draw a little picture of that, right? Where just to make sure you understand that we're talking about this transcription factor because it works by releasing arrested polymerase. So here's a heat shock gene. And it's got a promoter, and it's got an enhancer, and that enhancer is that heat shock response element. And in an unstressed cell, nothing's happening at the enhancer. However, it has already assembled a pre-initiation complex, and we have an arrested polymerase right there. And I won't draw in all the other proteins, but there's a bunch of other proteins hanging around there. And it's paused. It's ready to go. And what happens is we get the heat shock transcription factor to dimerize, like this. So they must have, HSF must have a DNA binding domain. And again, it's a weak one, but now we have 3, yay. Okay, it also must have a trimerization domain, a place where they can hold hands. And then it also has a transacting domain, because transcription factors have to have that. That's the business part that's going to do the job. And what does it do? It somehow physically interacts. The DNA folds over and it physically interacts with that RNA polymerase that says go. So that's the rate limiting step in the expression of these heat shock genes. And I mentioned before, most of our genes, more than half of human genes are regulated by having an arrested polymerase. And that transcription factor needs to bind to it and say, come on, let's get going. Okay, so that's our second example of this. So we go back to this and we say, right, in chapter 7, we're learning about gene expression, transcription regulation, and transcription factors. And then how do they do their job? They must increase a rate limiting step in the steps of transcribing. And there are some common ones. MyoD helps assemble the pre-initiation complex by recruiting TDP. Heat shock transcription factor helps release arrested polymerase on the heat shock genes. They're ready to go. And our third one is one we're going to talk a lot about, and it's remodeling chromatin. And our example is this estrogen receptor. It could be the testosterone receptor. They work the same way, and it remodels chromatin. So we'll talk about it a bunch. We'll have an assignment bringing this together, because we're looking at just the estrogen receptor as an example. We see a more complicated transcription factor than MyoD, and it's a lot we can learn. And so our book brings a lot of topics in that we can learn by just talking about the estrogen receptor. We'll use it as a specific example, but it teaches a lot of other things like that. For example, there are lots of transcription factors that remodel chromatin. They increase or decrease transcription by remodeling chromatin. There's a bunch of different ways to do it. Our book talks about too many different ways to do it in chapter 7. So we'll also zoom in on remodeling chromatin and make sure we understand good principles of what that is. Estrogen receptor does it one way, okay? So here's the chromatin. Chromatin is the DNA and all of the proteins bound to it. So transcription factors are part of chromatin, the DNA is part of chromatin, and that's what makes up chromosomes. When you say you're talking about a chromosome, you're talking about chromatin. And then chromatin is just, again, the DNA, okay? Like that, with all the proteins bound to it. And so we're super interested in this chromatin. The DNA molecules are really long, and they need to get packed into a cell. So part of chromatin always is a structural part of packing it in. And this shows that. This shows that there are these little blue balls of proteins that we're going to learn about, and the DNA wraps around them and helps it pack it, make it shorter. So you can see how the DNA here is really long, but it's in a smaller space because it's wrapped around these little blue balls like that, okay? And we see this term we need to learn. So when the estrogen receptor target genes are off, so in the absence of estrogen, the estrogen receptor is turning those genes off, and we'll learn about that. So we're talking about a female child that has these genes, and they're turned off. And they're packed away, and that's called heterochromatin. And that's found in female juveniles of all vertebrates, and also all male vertebrates. We have some awareness, right, that in humans, there's this kind of therapy where a person can get injections of estrogen or testosterone. And a male can have some female secondary sex characteristics develop. And that's because the estrogen response genes, the target genes, they're present in the males. They're just off. And the way they're off is this. They're packed away. And they can't express the gene. The RNA polymerase can't get there. Those pre-initiation complex proteins, they can't get there. You know, the example I always use is like, you know, you have some blankets you use in the winter and you fold them up, you put them away in the summer, you know. They're not being used now. It's literally physically packed away. And when the estrogen hormone shows up, we'll see the genes, estrogen receptor target genes, they get unfolded. And that's an example of remodeling this chromatin. So that's what chromatin remodeling is. We've got a structure to the chromatin, and we're going to change it. And we're going to open it up, and that's just going to give access to the pre-initiation complex and RNA polymerase to express the genes. So that's our third way. How do estrogen receptor transcription factors affect the rate-limiting step? The rate-limiting step is that in the absence of the estrogen hormone, the genes packed away. The rate-limiting step is you put those blankets away, and you have to get them out. You have to unfold them to use them. And that's just literally physically what's happening, okay? So none of this stuff is happening, right? It's not. It's all folded up and packed away. That's heterochromatin, and we need to unfold it, okay? So we're going to talk about one example of remodeling chromatin, and it's that one. It's going to be unfolding it. And so just to say this, right, we're talking about three major ways of regulating transcription, right? Assembling pre-initiation complex, releasing arrested polymerase, or remodeling chromatin. And I said our book talks a lot about remodeling chromatin. It's a big story, and there's a variety of ways to do it, and we're going to focus on three. So just to set that up, that as you read chapter 7, you don't have to learn so much about every one of those. Maybe there'll be a 1-2 point question. But the three kinds or examples of chromatin remodeling we'll learn about are these three, and we'll talk through them. Okay? And the first one is called histone acetylation. And that's what estrogen receptor does. So we'll talk about that first, and we'll talk about it in some detail. And then we'll quickly talk about nucleosome phasing and DNA methylation. So just to kind of give you some guidance on where we're heading. So remodeling chromatin, our favorite three ways are this, and all three of those could be examples of rate-limiting steps in gene expression. So histone acetylation. And here's, you can always just do a Wikipedia page to learn about chromatin remodeling. Wikipedia is great in the first couple of paragraphs anyway. Chromatin remodeling is the dynamic modification of chromatin architecture to allow the access of condensed genomic DNA to the regulatory transcription machinery proteins, and therefore control gene expression. Like that. So site remodeling is principally carried out by the first one, histone modification. In our case, it's this histone acetylation. And so these blue balls are called nucleosomes, and so let's just look at that. And the nucleosomes are made up of histones. So when we draw this DNA and we say, oh, the DNA is wrapped around this nucleosome, the nucleosome is that. So that's a nucleosome. And the nucleosome is made up of a set of 8 proteins. And the 8 proteins are 4 different proteins, and they are called histones. So the nucleosome is made up of histone proteins. And estrogen receptor functions by affecting these histones. And the way it works is that when the histones are packed, and we call that closed chromatin, heterochromatin, and this would be in juvenile female vertebrate. No estrogen, right? No estrogen, or because of male too, right? So it's packed away, closed chromatin. And what the estrogen receptor is doing is the estrogen receptor is holding, so there's the estrogen receptor over there, and the estrogen receptor is holding onto an enzyme. And that enzyme is a histone deacetylase. So that means it's an enzyme that's removing some acetyl groups from these histones. And when the histones are deacetylated, they fold up tighter. And so when I say, what does it mean when we fold up the blankets? It means we remove any acetyl groups, so they're covalently bonded, and we're just going to cut them off with this histone deacetylase, remove them, and when we do, the histones fold up tighter. So you could see this by saying this is deacetylated histones, and these are acetylated histones. And in the absence of estrogen, we'll see the estrogen receptor is holding on to a histone deacetylase, and then when we add the hormone, it lets go of that, and it binds to a histone acetylase. So the estrogen receptor affects chromatin remodeling by acetylating these histones in the nucleosomes and changing how they're packed. And that idea of the estrogen receptor being on a tether, right, is great, because it's like this little neighborhood that it's going to be affecting, right? So the histone receptor is bound, and it's just affecting all the histones within its reach on its tether. And so it's just unfolding this whole region into this open chromatin or euchromatin. Okay, so we want to look at that. Here's just an image of histones like that. There are 8 in the octamer, and then there's this linker one like that. That's great. Here we have added this histone acetylation. Open chromatin, euchromatin allows gene expression. Closed chromatin, heterochromatin represses gene expression. very physical and sensible, and we're getting there by just another statement like that about that. And what we're adding to our clay model is, we didn't talk about it, but the DNA is actually, the clay is wrapped around these nucleosomes all the time. And it can be wrapped around nucleosomes that fold up tightly, heterochromin are open, like that, okay? All right, so we're getting there. And so here's just an image of this. And so this would be the estrogen receptor, this would be estrogen, and these are the two situations. So in the absence of estrogen, we'd be up here, so the child, and then the teen, an adult, okay? And you can see the difference. We have the estrogen receptor here, no hormone bound, and when there's no hormone, it binds to this histone deacetylase transfer, or histone deacetylase is how a lot of people say it. When we add the hormone, it changes like that, okay? So we could draw that now and talk about what this estrogen receptor looks like. So now here's a gene, and we would just call this one of many estrogen target genes. Hundreds of target genes. And this is an enhancer right here, right? And most biologists, that's that enhancer. We've been using that. That's the term we're using on our course. This is a good enhancer, the estrogen receptor enhancer. It's a good enhancer to talk about that name enhancer. Because in the case of this enhancer, the protein that binds to it can either activate transcription or repress it. Because it's the estrogen receptor, and it activates or represses in a hormone-dependent manner. In the absence of hormone, it's not that the receptor is off, it's actually actively doing the other thing. In the absence of estrogen in juvenile female vertebrates or male vertebrates, the estrogen receptor is doing a job. It's repressing them. And so this is a beautiful example in molecular biology of a molecular switch. Where you don't want those genes on yet, you inherited them from your parents, you don't want them on, we're going to be actively repressing them and keeping them turned off, keeping the histones deacetylated so the nucleosomes fold up tightly into heterochromatin. And you can't possibly turn them on. But add estrogen to either males or juvenile females, and it switches. You turn from inactivating them to activating them. So let's just draw this estrogen receptor, because now we have to add more to its shape. Oh, and before we do that, I was going on this. Before we do that, let me just show you this. That people who study estrogen receptor, they just detest that name enhancer. And they like to call it a response element, right? Because that's a neutral term. So it's a response element. And although it binds to the estrogen receptor, it's really often referred to as an estrogen response element, because it's sensitive, responsive to estrogen. And estrogen target genes have an estrogen response, so it's abbreviated in ERE, like that. And that's where the estrogen receptor is going to bind. And obviously, the estrogen receptor must have a DNA binding domain. So the estrogen receptor has a DNA binding domain. Region that binds that DNA, that brings it, specifically to that response element, that enhancer. Then, we know that that it must also have, there's estrogen, right? The estrogen receptor must also have a ligand binding domain, right? Because ligand is a term for a small molecule that binds to a hormone and regulates it. So a ligand binding domain, right? Gotta have that, gotta have that. And that's like the light switch to activate or repress it. And then it has to have this too. We learned this today. It also has to have a trans-acting domain. So this has to have 3 domains. MyoD just has 2 domains, DNA binding and transacting. But here we have that 3rd domain to switch it on and off. All right? So now I'm just going to erase this and just draw that purple shape, because what we see is this. What we see is this, is that in the absence of estrogen, the DNA binding domain has a little bit different shape. And as a result, the transacting domain is shaped like that. So that's the DNA binding domain, the ligand binding domain, sorry if I misspoke, ligand binding domain, DNA binding domain, and transacting domain. And then, when we add estrogen, the ligand binding domain changes shape because it binds to that estrogen. And what happens is that the transacting domain also changes shape. So we have a shape change when it binds estrogen. So there's our estrogen binding tight to the ligand binding domain. And in the absence of the hormone, the transacting domain has a shape that binds to an enzyme that is a histone deacetylase. When estrogen binds, the shape of the ligand binding domain changes, and that causes a change in the transacting domain, and now the transacting domain lets go of the histone deacetylase, and it just binds to a different enzyme, and now it's bound to a histone acetylase. That's it. So a different enzyme, right? And it's an enzyme. So an enzyme is just going to catalyze, just hang on and catalyze. And it's just going in that region, right? It's tethered, right? It's tethered because the histone, because the enhancer is tethered to the estrogen receptor gene, and the estrogen receptor is bound to the enhancer, the response element, and it's holding on to one of these enzymes. So just holding in that neighborhood, and it's just removing all the histones like that when estrogen is present. So we just have a switch like that, and it's just the basis, right, for huge amounts of, who we are and who, vertebrate animals are. It's just that switch. So that's a great switch, and it's a great example of this chromatin remodeling, right? So it's just that, and like I said, the next chapter, chapter 16, actually starts talking about the histone receptor, because it's talking about cell signaling, and this is a type of cell signaling. So it's a big, big example. That's why we have that homework that's due a week from now. It's really kind of reviewing and trying to put the knowledge we have about the estrogen together in a package, because it's just such a significant example of a transcription factor and what it does like that. Okay, so questions about that? Yeah?
So we refer to the packed histones as heterochromatin. So once the deacetylase is released and it's bound to the acetylase, what do we refer? The unpacking, what does that refer to?
Yeah, it's so that I only think I said that word once, but the terms are heterochromatin is silent or closed chromatin, and then active chromatin is open chromatin or euchromatin.
And that's after the acetylase binds.
And yeah, okay. And another way to say that is these are acetylated histones in these nucleosomes, and these are deacetylated, like that. And other transcription factors do that too. Some, there are examples of that. Other transcription factors have other ways of remodeling the chromatin. For example, there are other types of histone modifications. The histones can get phosphorylated, they can get samoylated, they can get methylated. It's different than the DNA methylation. And those become really complicated because we can say this. In general, when histones are deacetylated, it's going to be repressing gene expression. And when they're acetylated, it's going to be increasing gene expression. And the book talks about other histone modifications, and it gets very complicated and confusing because of some of those modifications activate and sometimes they repress. So there's a big kind of blizzard of information about chromatin remodeling and the role of histones and transcription factors like that. And so we have a nice, clean, and just profoundly significant and satisfying example if we just say, let's focus on histone acetylation, the estrogen receptor, and how we open up chromatin in with the presence of the estrogen hormone. That's what it's doing. Yeah, okay? So questions about that? Yeah.
I had a question about the HFF
Yep.
monomer. Is it the trimer that binds to the HSRE?
Yes.
Okay, and that's after the monomers are denatured?
Yeah, slightly
From the trimer?
denatured, so it exposes that trimerization domain that they bind together. And the way to remember that is it's like a three-handed climber, right? So, and you know, it's kind of a brutal analogy, but you know, one-handed climber has a hard time because they don't have that extra hand to hold on to, right? And if one hand lets go, boom, they're gonna fall, right? But a three-handed climber can let go with two hands. So, you know, even though the hands are low affinity in each of the heat shock transcription factors monomers, they're effectively holding on really tightly, like that.
What induces the stress to denature the monomers?
Yeah, so it could be heat,
Okay.
right? And so that's why I mentioned it's like a thermostat. And a thermostat works by having a little bit of metal in it that is temperature sensitive, and it's just bending with the temperature. And when it bends enough, it touches and closes a circuit, and you get the heater on, like that. So it is like a little thermostat. So it's a satisfying example because it's trying to monitor the protein denaturation status. And it's just a little bit sensitive to that. It doesn't completely unfold. It unfolds a little bit, exposing the trimerization domain, you know, boom, like that, you know. So very physical, very sensible, kind of satisfying stories like that, yep.
Can I ask a question about the first part
Yep.
of that too? The MyoD binding domain, the DNA binding domain binds to the enhancer. Does that same domain also bind to TBP, or is it
No,
the
so
transacting domain?
yeah, so the transcription factor is going to have a DNA binding domain to bind to the enhancer, and then it's going to have a transacting domain that's going to do whatever it is that's going to, you know, increase the rate limiting step. So there must be a transacting domain on MyoD that binds TBP and just like get, you know, stay here or get here. Yep.
Great, thank you.
Other questions people have about anything at all? Yeah, and so you see all these in pieces, all this stuff is like, yep, I could draw that out, right? Draw that out, that makes sense. And it's weird, you know, because you're like, is it more complicated than that? Well, you know, you can add some details, and the book makes it more complicated by diving into some of those other examples. But, you know, we should build a framework where you're like, poosh. The problem is, even just with what we talk about in class, it's a lot like that. Yep, okay. So, chromatin and histone acetylation, like that, okay? Great. So here's just this little image of, you know, the actual, you know, covalent bond of the acetyl groups like that, you know, just some details on like that, right? Sex reassignment therapy. Here's another example we want to talk about, as we talk about chromatin remodeling, we want to talk about ester receptor and this. So there's, and because it's now an example, it fits into ester receptor, fits in chromatin remodeling, fits into histone acetylation, but it's about cancer biology. And so that's why our book goes there next, is because we're going to go into this section about cancer, right? And so the example is breast cancer, okay? So if a person has a lump or a bump anywhere in their body, the doctor will say, you know, we're going to take a little of that tissue out, and we're going to do a biopsy, take a little tissue, and we're going to check, and we'll talk about that. What do the doctors and the helpers in the clinical lab, what do they look at? So today, now, if a person has a lumbar bump in their breast, the doctors will look, and what they'll do is they'll analyze if those cells are expressing the estrogen receptor. So they're just detecting it. They could look for the estrogen receptor RNA or the estrogen receptor protein. And if they find it in that lump, bump, they say it's ER positive. That's what that stands for, like that. In the United States, about 70% of breast cancer tumors are ER positive. That means 30% are not. And there's a good chance that if a person is diagnosed with breast cancer, the doctor will even say to them, this is an ER positive breast cancer, or this is an ER negative breast cancer, because there's a drug called tamoxifen that's really good on ER positive breast cancers and does nothing in an ER negative breast cancer. And the reason it is because tamoxifen binds to the estrogen receptor. So it only helps if the estrogen receptor is present, like that. And tamoxifen is an antagonist of estrogen. And so an antagonist means it does the same type of thing as estrogen, except it does the opposite. And for the word antagonist to be used, you have to have it be binding directly to the receptor. And so that's what tamoxifen does. And we could just draw tamoxifen like this, where tamoxifen binds and it doesn't change the shape. So that's what tamoxifen does. Now it's spelled on the other one, you can see it spelled out there. And so you see these ER positive breast cancers are growing, and they're growing in an estrogen receptor dependent way. Estrogen can stimulate growth of the breast during puberty, right? Like that, of the cells, and this is going to just block that. And it's going to make that cancer stop growing because the cancer is growing dependent on estrogen. And you can see what's happening is that not only are we saying don't work estrogen, we're making it do the opposite. We're like, let's shut this thing down. And so because of the mechanism, this antagonist, this opponent to the estrogen, is very effective at stopping those breast cancers. But it's useless in an ER negative breast cancer. And that's, another, okay, that makes sense. It's a fact. And that's tough, right? Because a patient could be ER positive and take this tamoxifen, and it doesn't cure the cancer, but it puts it in remission very well, like that. And that gives the doctors other time, time to choose, additional strategies, stuff like that, right? So we're not talking about a cure, but we're talking about a very effective drug for ER positive cancers, like that, okay? And we're talking about right now because you can see, oh, wow, yeah, good mechanism, right? Because it actually shuts down the cells that are growing, like that, okay? So when we're talking about it, what then goes on is just that, and I have a couple of slides about tamoxifen because it's been used for years to do this like that, and it's very effective. And there's some other drugs, and it has a trade name too, like that's not the name they sell it by and stuff like that necessarily. So lots and lots of people in the United States have taken tamoxifen when they were diagnosed with breast cancer, and it was very helpful. Now, just recognize that then there's a set of people who are ER negative, and they can't use tamoxifen. So what happened years ago is that scientists started working on these ER negative breast cancers. What's making them grow like that? And it turns out that they found out some of them were dependent on another receptor and another growth factor, and it's the glucocorticoid receptor. And so they found a drug that would stick to that. And so in those ER negative ones, they would find a way to treat some of those patients with another drug that would only be useful for patients that were ER positive to that. Yep?
Does tamoxifen have a higher affinity than estrogen?
Yeah, that's a good question, and the answer is, I mean, I don't know off the top of my head, but, well, I have known that, I think I might even have it on a slide, so I'm gonna say that what happens with tamoxifen is that I think it gets modified when it gets into the cell, and that modified version does have a higher affinity, so it is like a competitive inhibitor type of situation, yeah. Yeah, I'm just trying to remember, because I know that for sure. But I think that's the case. And so they found, ER negative, but then they found some of the ER negative could be treated another way. So then there are some patients that are ER double negative, right? Negative for estrogen receptor expression, negative for this other receptor. Let's try to treat those patients, right? And they found another growth factor and another receptor, and so there are now a few patients with breast cancer who are said to be triple negative. That's how they just say it, triple negative. They got 3 good ways of treating almost, the vast majority of breast cancer, at least, not to cure, but to get a really good help, And so there's a triple negative. And I mention that just because there is medical research at UNE who's developed a drug to treat ER negative breast cancer patients, so that research is ongoing, and that's typical that we'll learn about in modern cancer biology, is many cancers we found a drug that will specifically help that type of cancer, not just breast cancer, but a certain type of breast cancer. And so in many cases now, when a patient gets tested, the lump or bump gets biopsied, and they look at it, they'll come back with some detailed understanding of what's causing this cancer and whether or not they have a drug that works well on it, or some kind of treatment like that, okay? So just diving into that, leaning into that a little bit, and here's, tamoxifen bound to the ligand binding domain of estrogen receptor, and the question is about this kind of competition between estrogen and tamoxifen finding, and again, many people have taken it, so I just put that on there because it has some of these brand names that are more, maybe more familiar like that, and just more information about that. So it was approved in 1998, tamoxifen, and treat millions of people like that, right? Okay, so that's one example, and we'll go into that because we want to talk about cancer biology. Here's another example of an estrogen receptor story to tuck this in. So everybody's heard that BPA is in plastic, and you don't want to have it in plastic. BPA does this too. BPA binds to the estrogen receptor and is called an agonist. And so those are 2 important words for us. Antagonist is something that binds directly to a receptor, directly to a protein, and inhibits it. It does the opposite of what the normal hormone or molecule would do. An agonist, and you've heard the word antagonist, right? I'm antagonizing you, I'm opposing you. You might not have heard that root word agonist, but an agonist is a molecule that binds to the same target receptor, the estrogen receptor in this case, and it activates it. And that's literally what BPA does. So if we go back to this drawing here, sorry, no, still haven't gotten that down. Wow. So this is what the BPA would do, right? Is it mimics that estrogen like that, and so we've got it binding here like that, and you don't need estrogen. So who does BPA impact? Well, especially males and juvenile females, because it replaces estrogen like that. And a good way to remember that is years ago, Maine had a governor named Paul LePage, and he gave a press conference once, and he's a guy, and he said, I'm not worried about BPA and plastic in Maine. And as I understand it, it would just cause women to grow little mustaches. And none of the news reporters, I guess, knew molecular biology because, right, the real thing is, well, he should be concerned, right, because it would cause him to grow breasts. And it would be interesting, right, you know, because you have that a lot, where a lot of people are like, I'm not worried about this, it's not going to affect me. And if you said, excuse me, Governor. So it's just a funny way to remember it. You know, I'm not making any political comments other than just, you know, we have politicians, they don't understand the science. We have news reporters, they don't understand the science, and so they're talking about it, and you're like, I don't know, you know, you might be a little bit worried, like that. So it's an agonist, and I put up here a fish because BPA doesn't just affect humans, it affects all vertebrates that use, you know, estrogen as a sex differentiation hormone. So there's lots of fish out in the wild now that people are seeing that are called intersex. They're sterile, and they're supposed to be males, but they got exposed to too much of something like BPA, because it turns out BPA is not the only molecule that do that. And those are called endocrine disruptors. Endocrine means hormone, and they're disrupting the normal hormone development. So estrogen receptor, lots and lots of, you know, important parts of our story. And, you know, we have added that, you know, back here, right, in our list. We added it, you know, a couple weeks ago or whatever, estrogen receptor. It's just fascinating, all the different ways it works, right? And we want to put some of that knowledge together in a package in that drawing assignment for a week from today, like that, okay? I mentioned that it's a member of a gene family. We'll talk about that gene family some. The gene family is called nuclear receptors, and nuclear receptors have a good name because they're receptors that function in the nucleus. So these are all 48 of them are transcription factors that function in the nucleus. Estrogen receptor, testosterone receptor, there's a whole bunch of them that are really important to our health and vertebrate health, like that, okay? Great. So that's all tucked into that context of learning about estrogen receptor and learning about it in chapter 7, and how does it work? How does it regulate transcription? And it regulates it that cool way, okay? So here's just a picture of that idea, right? DNA binding domain, ligand binding domain, transacting domain in an estrogen receptor or any of these nuclear receptors. Then just to mention a little bit more, it's been studied and studied and studied. Here's the DNA binding domain of the estrogen receptor, and it has a very cute binding domain because you can bind to DNA, and it has fingers, and they're called zinc fingers because zinc is coordinating binding the amino acids to make that correct shape. So we can just add to this drawing that this DNA binding domain is just got, it's called a zinc finger. And the thing I always think about is in baseball, the different players have different kinds of gloves, mitts, to catch things. And that's true for transcription factors. The DNA binding domain is kind of in some different styles. And there's a whole bunch of transcription factors that have a zinc finger style, a certain kind of binding glove, like that. And so people just like to point that out. Oh, that's a receptor, yeah, it's got zinc fingers, you know, that's how it binds. So we know that, you know, the DNA binding domain has a very, very specific shape. It's always got zinc bound in there. That's part of that structure, just to sort of point that out as a fact of that story, like that. Because people know a ton about these nuclear receptors and how they bind. So just a couple of images about that for you to get members. I go into that a little bit more in that homework assignment. And here's just a phylogenetic tree of all 48 of those. It's just a very, very, so estrogen receptor, testosterone receptor, thyroid hormone receptor, glucocorticoid receptor, vitamin D3 receptor, this receptor called PPAR, and just fantastically significant stories in our health, 48 of them, like that. And they're paralogs, like we learned about, but there are really diverse families, so like to call it a gene family that's a super family like that. So they evolved to have slightly different functions from each other. Yep. All right. So I'll just skip over those because again, I go into some of those in a little more detail. So now, we're doing great, and we have a few more minutes, and we're just going to introduce this next type of chromatin remodeling. So remember, chapter 7 has too many examples of chromatin remodeling, and it's confusing So we're going to learn especially about 3, to have you guiding through there, and maybe you'll learn some more. That would be great. So the first one is that histone acetylation, and our example is the estrogen receptor. There are lots of other receptors that do it that way too, because they're remodeling chromatin in order to increase the rate of transcription. So here's our second example of chromatin remodeling. And it's an example that's really significant that I don't think our book ever mentions, and it's great to mention because it fits into an existing example in a really satisfying way. And our example is back to that heat shock transcription factor, and we're going to add another transcription factor that's got another great name. It's called GAGA factor, right? And so it's going to look like this, where here's one of these heat shock genes. And it's already got an arrested polymerase on it, right? Just part way down it, but it's stuck, so an arrested polymerase. We learned about that. And then it's got this heat shock trimer bound here. Activating gene expression. And somehow that DNA is bending over and the transactin domain of HSF is making contact with the arrested polymerase and making it go. In this story, there's another binding site involved, and it's here. And if you look at the DNA sequence, it's like G, A, G, A, G. It's just got a bunch of G's and A's. And there's a protein that binds to it, and it's our new protein, and it's called GAGA factor. And it is remodeling the chromatin. And the GAGA factor is always present. And what it's doing is it's blocking nucleosomes from binding right there. So if we drew those nucleosomes, and those are those histone balls, the DNA is wrapped around it, they're all lined up like this. And normally those nucleosomes are just scattered around. They could be here or there, they could be different sites on different genes. And what GAGA factor is doing is it's just doing that so we always have this space ready, right, for that heat shock transcription factor. So it's another really satisfying example, because when we trimerize that heat shock transcription factor, we want it to be able to find that heat shock response element now. And so GAGA is always hanging out there, and it's just blocking nucleosomes from binding that site and slowing the story down. And that's called nucleosome phasing, because the nucleosomes in every cell are at the same positions on every heat shock gene, because they're always being phased or positioned to make sure they're not blocking the HSRE. So that's as simple as that. GAGA factor, it's a memorable one. It fits into an existing example. It's a satisfying example that's there to help be ready to go. And it's called phasing nucleosomes, and what we're doing is we're just positioning the nucleosomes to make sure we have space ready for what we want to do, there. And it's actually quite common in a variety of different ways that we remodel chromosomes by positioning these nucleosomes to make sure they're not in the way, like that. So that's that one. We'll quickly cover DNA methylation after that, and then we'll be kind of close to wrapping it up because we have another assignment that is focused on chapter 7 also. Yep. All right. So that's where we're at.